So, hello everyone, I'm Nick James, and I'm very excited to introduce our first Newman speaker of 2021, Dr. Joel Boerckel. Joel received his PhD from Georgia Tech in mechanical engineering, and during his study developed an immense interest in developmental biology. He went on to Cleveland Clinic, where he received an F-32 award as a postdoctoral fellow, and in 2014 became an assistant professor at Notre Dame in aerospace and mechanical engineering. Since 2017, Joel has been an assistant professor at the University of Pennsylvania with joint appointments in orthopedic surgery and bioengineering. Joel has become a very accomplished young investigator in the last 10 years, even receiving an ORS Young Investigator Award in 2019. And his lab's research is focused on understanding the role of mechanical stimuli in embryonic development, and was actually the first to identify the roles of YAP and TAZ in bone development. They then applied their insight to regenerative medicine techniques. And lastly, while I don't know Joel super well yet, from what I understand, we both enjoy a nice Belgian beer from time to time. All right, Joel, please go ahead.
Thanks so much, Nick. Okay, so today I want to tell you all about some of my lab's work in mechanobiology of development and regeneration. I want to thank Hani and Nick for the kind invitation to be here. I love the community that you all have. It's been such a pleasure to talk to so many people, and I look forward to more conversations throughout the day. I wish we could be all together in person, but we're gonna try to get through this together. So I'm gonna need some help from all of you to make this happen. Okay, so today's talk is going to be what I call a choose your own adventure. When I was a kid, I loved these choose your own adventure books by this guy, RA Montgomery. Now, if you were a kid or you were the parent of a child in the 1980s, you're probably familiar with these books. But some of you who are in a younger generation may be more familiar with Bandersnatch. My kids right now are really into this show called You vs. Wild. And of course, what all these have in common is that they are choose your own adventures. So here's the map for this particular choose your own adventure book. And in this book, the protagonist encounters this monster and has to decide whether to fight the monster or run away. And as the reader, you decide whether you fight the monster and go to page 27 or run away and go to page 135, right? So what we're going to do today in this talk is you all are going to decide what we talk about. And the way this is going to work is I'm going to have polls set up in Zoom, and you are going to choose a blue option or a red option. Okay, and I want to start with a relatively low risk choice here to get us started. So the question is, who would you like to introduce today's talk? Do you want to hear from a donkey killer, this guy with this lovely beard, or this pair of thieves? So let me launch the poll, and everyone gets to choose red for the pair of thieves or blue for the donkey killer. It is neck and neck, everybody. I think we're coming to the end. A couple more people voting. All right, I'm going to end the poll and share the results. So red wins it by a nose with 53% of the vote. Okay, so let's go to red. So you chose the pair of thieves, and I love this story. This is a picture of these two guys. Their names were Albert Ebenezer and Ebenezer Albert Fox. And Albert and Ebenezer were criminals. They were thieves and poachers who lived in the forests around London at the turn of the 20th century. And they had a criminal advantage, which is that they were identical twins. So one of them would go off thieving and poaching, and the other one would go to various pubs and introduce himself alternately as Albert or as Ebenezer, and thereby establish an alibi for the both of them. And this worked great until this guy came along. Edward Henry was a commissioner with the London police, and he had a hobby of keeping up with the scientific literature. And one day he noticed, or he found this report about this new finding that he thought might help him catch these guys. And this, of course, was the fingerprint, or as I like to call it, mechanical control of morphogenesis. And what I love about this example is that we can actually solve these field equations. If we know the boundary conditions, we can predict the patterns of the whorls and the swirls in the finger digit because of the mechanics, right? And this is why monozygotic twins can be differentiated by their fingerprints because of the forces that they exert in the womb and against one another as they jostle, produce different boundary conditions that lead to different patterns. But the message that I want everyone to take away from this is that what this says is that mechanical forces are important in the process of development. Now, I like to share this quote from one of my heroes. This is from Victor Hamburger, who's one of the fathers of developmental biology. And he said this: "Our real teacher has been and still is the embryo, who is incidentally the only teacher who is always right." So taking inspiration from the fingerprint and from Professor Hamburger, my lab is interested in what we call reverse engineering development. First, we are interested in understanding mechanobiology. How is it that cells sense and respond to mechanical forces? And we're interested in then applying these findings to developmental biology and trying to understand how do these mechanical cues influence development. And I'm going to tell you some stories today about the development of the skeleton, but we're also interested in vascular development and other things. And then we take these ideas and try to apply them to the problem of regeneration. So can we develop tissue engineering strategies that recapitulate or say reverse engineer the process of development? Okay, so let's begin by talking a little bit about endochondral bone development. I think this is a really amazing and beautiful process. We start with an embryo and here I'm showing a mouse embryo at 12.5 days of development. And this embryo looks just like any other embryo you've ever seen. But within 1 week, it turns into a mouse with a skeleton. And I think this is amazing. Now, if you're a zebrafish researcher or something, you're probably laugh at these timescales, but I think it's really remarkable. And the way that this works is that you start off with a limb bud that is a condensation of mesenchymal progenitor cells that have the capacity to become other cell types. And what's really amazing is that they don't become bone, right? The goal here is to make a bone, but these cells don't become bone. First, they become cartilage. And they form a cartilage template that gets remodeled and replaced with bone through this process of endochondral ossification. And there's various stages in this process, and each of these stages require decisions. We have chondrocytes that have to go into hypertrophy. We have vascular invasion and recruitment of osteoblast progenitors. We have remodeling and resorption. And each of these stages requires cellular decision making. So when I started my lab, we sat down and we looked at all of these transcription factors that are important in making these decisions. And it was a hopeless mess, right? There's so many transcription factors and they're connected in so many remarkable ways. And so we took a step back and we said, well, what is it that could regulate these decision makers in the cell? How do we coordinate these? And it turns out that many of these transcription factors that are involved in the process of skeletal development have these protein-protein interaction domains, these PPXY motifs that allow them to bind to other proteins that have what are called WW domains. And there are two proteins that are particularly interesting to us that have these WW domains and can bind to a whole variety of transcription factors and function to regulate transcription factor activity. So these are just some cartoons of the structure of these proteins, YAP and TAZ. YAP and TAZ are what we call transcriptional regulators. They cannot bind DNA by themselves. So they don't have DNA binding domains, but they have these protein-protein interaction domains. So they can bind the TEAD family of transcription factors, the WW PPXY interactions here and these PDZ binding domain. But they also have a transcription activation domain, and this allows them to regulate the transcriptional activity of other transcription factors. Let's see. Okay, now the other really interesting thing about YAP and TAZ is that they can be activated by mechanical cues. So anything that can cause tension of the cytoskeleton, for example, fluid shear stress over the cell, or strain of the extracellular matrix, or the mechanical properties of the extracellular matrix, in response to these mechanical insults, the cytoskeleton will respond, and this results in nuclear localization of YAP and TAZ. So remember, YAP and TAZ can't bind DNA by themselves, so they have to bind to other transcription factors like the TEAD family. And together they form a transcription factor complex that can regulate gene expression. But if they're sequestered in the cytosol, then they're held away from their transcriptional partners, and you don't get YAP-TAZ dependent gene expression. So all of this together said that YAP and TAZ can bind to maybe these transcription factors that are important in skeletal development, and YAP and TAZ can control transcription factor activity. So we thought, well, maybe YAP and TAZ play a role in osteogenesis. And it turns out that we had waded ourselves into a 15-year-old controversy. So the first paper on YAP in osteogenesis showed that YAP can bind to this master osteogenic transcription factor RUNX2. It binds to RUNX2, sequesters it from the DNA, and prevents RUNX2 dependent transcription and osteogenesis. So YAP is an inhibitor of osteogenesis. The following year, TAZ was identified as a transcriptional co-activator of RUNX2, binds to RUNX2, drives RUNX2 dependent transcription and osteogenesis. And every year since then, there have been new papers of in vitro studies showing that YAP and TAZ can either co-activate or co-repress, have convergent or divergent functions. You can flip a coin. And so we saw all these in vitro papers, and we thought the only way that we're going to be able to make sense of this system is to study this in a physiologic context, and to try to tackle these two together. And so we took this approach. We used the floxed mice, in which YAP and TAZ, a portion of the gene is floxed with these locations where this enzyme, Cre recombinase, can come in and delete that part of the gene, rendering the product non-functional. So we can delete YAP and TAZ conditionally in the cells that express this ostrix that drives the enzyme. We used a breeding strategy that would give us these different outcomes. We can have mice that are wild type for YAP and wild type for TAZ. Remember, each gene has 2 alleles, so we have 4 total copies, right? At the other end of the spectrum, we have mice that are completely knocked out of YAP. So both alleles of YAP are knocked out and both alleles of TAZ are knocked out. And in between, we have these varying allele dosage. So 2 heterozygotes, a knockout and a het, or a het and a knockout, etc. So we can think of this as decreasing allele dosage from left to right. And what we found in these skeletal lineage cells is that if you delete both copies of both YAP and TAZ, so double homozygous knockouts, the mice died shortly after birth. And this is remarkable because we've targeted YAP and TAZ just to the bone lineage cells, and the mice are dying. But a single copy of YAP or a single copy of TAZ was sufficient to rescue this perinatal lethality. So the question is, why? Right? Let's try to figure out why they died. Now, I'm going to have you put yourselves in my shoes as a new assistant professor with getting a phenotype, right? We're ready to do something, and we gotta figure out what to do next. So it's up to you. Should we figure out how cells learn to make these kinds of decisions, take a cell biology approach, or should we take a developmental biology approach and look at the embryos? So let me launch choice poll number 2, and everyone gets to pick red or blue. All right, we're settling in here. I'm going to end the poll and share the results. It looks like red wins 54 to 46. Okay, so let's go look at the embryos. So the story I want to tell you now is ongoing work from a PhD student in my lab, Joe Collins. And here we're going to focus on the roles of YAP and TAZ in embryonic development. Now, Joe began this, this study by first asking, what are the cells that are at play here? Who are the cells we should care about? And so in collaboration with Nat Diamond, who developed these reporter mice, we can look at the different cell types in the embryonic limb. So here we're looking at the femur and the tibia, and the cells that express collagen 2 express this transgene that drives expression of a cyan fluorescent protein. So all of the collagen 2 expressing chondrocytes are blue. The collagen 10 expressing hypertrophic chondrocytes are red, and the collagen 1 expressing osteoblasts are yellow. We can zoom in on the primary ossification center here, and you can see these different cell types, right? So we have at the edges, we have what's called the bone collar, where intramembranous ossification causes differentiation of these periosteal, perichondrial cells into osteoblasts. At the center, we have the primary ossification center, where chondrocytes enter hypertrophy, and then we get endochondral ossification. Okay, so we can use imaging approaches to quantify the distribution of these cells, and you can see that collagen 2 positive cells drop off in the middle, but are high at the outsides. We can see these 2 peaks in collagen 10 positive cells as the 2 growth plates begin to separate apart. And we see this peak in collagen 1 staining right at this time point, which is embryonic day 15.5, is primarily in the bone collar. Now, I told you earlier that YAP and TAZ are regulated by the cytoskeleton. So tension in the cytoskeleton can drive YAP and TAZ into the nucleus. And so we looked at the actin cytoskeleton by staining phalloidin. And here we see, right at the initiation of the primary ossification center, we see this peak in phalloidin staining. And this led us to ask, okay, what is it that YAP and TAZ are doing in the embryonic limb? So we can take these mice and stain for YAP and TAZ, and we find that YAP and TAZ are robustly expressed essentially in all of these cells that we're looking at here: the hypertrophic chondrocytes, the osteoblasts in the bone collar, etc. So the approach that we took is to use this Ostrix-1 Cre. And here these are co-labeled with a GFP, and you can see that Ostrix Cre is expressed in all of the same cells that YAP and TAZ are robustly expressed in. Okay, so our approach here is to say YAP and TAZ are mostly expressed in these cells. Let's knock them out of these cells and see what happens. All right, so if we do an alkaline phosphatase activity stain, we can see this is sort of a marker of osteoblastogenesis and osteoblast activity. We can see lots of osteoblast activity in the bone collar. And here in the primary ossification center, we're starting to see bone formation at embryonic day 17.5. But if we look at these knockouts, the alkaline phosphatase activity in the bone collar is still there, right? It's not too different. But in the primary ossification center, where we get this endochondral ossification, there's a dramatic defect, and we can quantify this over a number of different embryos. So this suggests that YAP and TAZ are having an effect, maybe especially in this endochondral ossification mode. So we went back and looked at the phalloidin staining here at 15.5. And you see, just as we saw in the reporter mice, there's this peak in phalloidin staining that shows up at the center of the initiating primary ossification center. But this disappears in the knockouts. And if we look 2 days later, as the primary ossification center is expanding, we see this plateau of high phalloidin staining, F-actin, in the wild types, and it's suppressed in the knockouts. And if we quantify the cellular density in this primary ossification center, there's a significant decrease in the number of cells inside the POC. So this told us that there's a difference in the number of cells, but is it because there's a decrease in the number of cells from which we've knocked out YAP and TAZ? So here we use the Ostrix Cree GFP as our wild type to mark those cells with the GFP. And inside these dotted lines, you see the primary ossification center. And here we have a significant decrease in the number of Ostrix positive osteoprogenitor cells that mobilize to enable endochondral ossification. Now, endochondral ossification, by definition, has to start in the growth plate, in the cartilage. And so it's also important that Ostrix is not only expressed in the osteoprogenitor cells, but is also expressed in hypertrophic chondrocytes. So during endochondral ossification, the Ostrix turns on in what we call this maturation phase. So we've broken the hypertrophic cartilage down into 3 regions. It's entry, the maturation zone, and a remodeling zone, where the hypertrophic chondrocytes and their matrix have to be degraded to enable this transition to bone formation. Okay, and here at this remodeling line, we have this beautiful organization of cartilage degrading capillaries that are responsible for producing enzymes to degrade the extracellular matrix. So here I'm showing a collagen 10 immunostain. So this is collagen 10, but we're not looking at the collagen 10 expressing cells, we're looking at the collagen 10 itself. And in the wild type, you can see this nice, so here this is the humerus, and we're looking here at the edge of the growth plate. This is the transition zone between the primary ossification center and the growth plate. And it's this beautiful straight line. And it's always this beautiful straight line. And we can label our different regions: the entry zone, where collagen 10 is largely intracellular; the maturation zone, where those chondrocytes are spitting out collagen 10 into the matrix; and the remodeling zone, where the collagen 10 is essentially all in the matrix. Now, if we look at our YAP-TAZ knockouts, we find there's an elongation of the maturation zone. They enter hypertrophy at a normal place, right? The positioning here is identical, but their maturation is extended, and there's a dramatic defect in the remodeling. And this takes on a real characteristic shape. The hypertrophic chondrocytes, instead of ending at this beautiful straight line, they have a cone shape. And this suggests that there's a defect not only in the maturation of the hypertrophic chondrocytes, but also their remodeling, as they get remodeled from the outside in. Now, the remodeling of the chondrocytes happens in concert with blood vessel invasion and vessel-mediated degradation of the matrix. So we looked at endomucin, we used endomucin to mark the endothelium. And here we can quantify vessel areas throughout the tissue, right? We see that in the knockouts, there's a significant decrease in vessel area. But let's zoom in on this region where the blood vessels are responsible for degrading the cartilage. And you can see here this cone shape of the hypertrophic zone as the blood vessels are degrading from the outside in, but don't reach in the middle. And the organization of these vessels is also defective. So I've skipped over a bunch of observations that we could make here, but I want to make some conclusions. We find that YAP and TAZ have roles in osteogenesis, but especially in the mobilization of osteoprogenitor cells. They deleting yapentase leads to defects in hypertrophic chondrocyte maturation and remodeling, in part through this effect on angiogenesis and osteogenic angiogenic coupling. Okay, so this says that yapentase are really important in the development of the skeleton. But our goal here was to figure out why the mice died, right? So if we look right at birth, what we find, here's the skeletal preparations of the P0 skeletons, the rib cage and the hind limb, stained with Alcian blue and alizarin red. And you can see that the femurs are broken and the rib cage is broken in the knockouts. And it's these rib cage defects that cause that perinatal lethality. So the mice, because of malformation and fracture of the rib cage, are unable to breathe after birth, and they asphyxiate. And the reason for these defects in the bone is because of these embryonic defects in all of these processes that we talked about here that lead to bones that are brittle and have low fracture toughness. Okay, so now we know that yapentase are important in bone development, but we have some options. We could continue on looking at embryos and try to see how these mechanical forces can influence bone formation in the embryo, or we can use this information to try to recapitulate the process of bone development for tissue regeneration. So these are our two options. Let me go to launch the poll, and everyone can choose red for development mimetic regeneration, or blue or for mechanical control of morphogenesis. All right, I'm going to end the poll and share the results. It looks like blue has won with 57% of the vote. Okay, so let's go here. We want to see how mechanical forces regulate bone development. So this is work from Joe Collins and Yasemin Mahar, who are both PhD students in my lab. And here we're going to try to figure out how does this mechanobiology influence the development of the skeleton. And it turns out this is a very old problem. A little while back, I went back to reread Julius Wolff's famous book on the law of bone transformation, where we got Wolff's law. And he's famous because he came up with this idea that the morphology of the trabecular bone is influenced by the mechanical forces on it. But he actually spends more time in this book on how bone morphogenesis happens than he does on trabecular bone adaptation, which I think is really, really wonderful. Now, more recently, other very important people have studied this problem. So here's a picture of a reprint I have from a paper by Victor Hamburger, that same guy that I quoted from at the beginning of the talk. He was a developmental biologist interested and studied chick embryo development. And the beauty of the chick model is that you can culture chick embryos in an incubator, in a dish, and then you can do things to them during development. And so he was interested in how nerve innervation influenced the development of the muscles. So he would take chick embryos and cut the nerves and look at their muscle development. But one of the things that he noticed during these experiments in the 1930s is that when he cut the nerves that enabled muscle contraction, the bones didn't develop properly. So this suggests that these mechanical forces exerted in the embryo during development are important for the development of the skeleton. Now, you may ask if this is important in humans. Here's some ultrasound videos that were taken by our collaborator, Neve Nolan. These are high resolution ultrasound videos of a human baby in the womb from 20 to 35 weeks of development. And Neve and at the time her PhD student Stefan solved the complicated inverse dynamics problem to figure out how do you go from boundary conditions of baby kicking in the womb to local stresses and strains on the developing limbs. And what she found was really remarkable. It was that there's a concentration of local stresses at the locations where, when there are conditions that limit fetal movement, you have developmental dysplasia. So here at the hip are high stresses. If you have, say, an insufficiency in amniotic fluid, the womb constricts around the baby can't move as much, and you get developmental dysplasias that occur right at these same locations. So it suggests that these mechanical forces are actually really important in human development. Now we want to study why, right? What are the cellular and molecular mechanisms that enable this process? So in collaboration with Neve, through a collaborative exchange grant from the ORS, we implemented her bioreactor system, where you can take embryonic limbs, explant them from the embryo, and culture them under dynamic mechanical loading conditions in a bioreactor and keep them alive. So here's a cartoon of this process. We have these embryonic limbs that were taken at embryonic day 15.5. So if you remember from the histology, that's right at the beginning of primary ossification. And we pin them at the hip and then put the feet against this support. And then the bioreactor will compress this foam on which they're sitting, which with a 2 millimeter displacement of the foam results in a 14-degree flexion at the knee. And so here, as you can see in this video, we'll do dynamic compression, which causes bending at the knee of about 14 degrees for 2 hours, 3 times per day. And we did this in the experiments that I'll show you for either 5 or 6 days of mechanical stimulation. And they were compared with static controls. So here's some optical projection tomography images. You can see the femur at the top, the tibia, and the fibula. And if you remember, the fibula is sort of shielded from these mechanical loads by the tibia, and this will be important in a minute. But what we see after 5 days of dynamic loading is that it looks like there's more bone in the dynamic loaded bones than there is in the static. So we can quantify this bone formation in a number of different ways, but what we find is that both in the femur and the tibia, there's mechanical loading causes an increase in the length of this bone. I call this the bone collar length, but we don't have the resolution right here to differentiate between endochondral or bone collar bone formation. But importantly, in the fibula, which was stress shielded by the tibia, we don't see an effect of loading. Now, the question is, do YAP and TAS mediate this mechanoregulation? And so I just have one sort of small piece of data to show you, though we're working on some more that I'm really excited about. So here we're going to take a drug-based approach, and we're going to use this drug verteporfin, which interferes with the interaction between YAP and TAS and the TEAD transcription factors. And this complex is responsible for driving expression of this gene CTGF. And so when we treat cells with verteporfin, we can reduce the expression of CTGF and reduce the transcriptional activity of these TEAD transcription factors. So we're going to use this drug in the bioreactor system. And what we found was that under dynamic loading in DMSO-treated embryonic limbs, we have an increase in alkaline phosphatase activity. But when we treat these with verteporfin, we don't get this increase in ALP. And we can quantify this here, an increase by dynamic loading, but is completely abrogated by the verteporfin. So this suggests that YAP and TAS play a role in transducing these mechanical signals to influence bone morphogenesis. Okay, so so far we've found that YAP and TAS are important in bone development. and that they may play a role in mediating mechanical regulation of bone formation in the embryo, or at least in embryonic explants. But what do we want to do next? Do we want to try to figure out how cells learn from these forces, or use these forces for regeneration? All right, let me launch a poll. And everyone has an opportunity to vote.
Hey, Joel, real quick,
Yes.
while we're answering this poll, we have one question
Great.
from Fong Nguyen. He's wondering how you kept the explant limbs alive for 5 days in vitro.
Great. Okay, this is great. So it turns out that if you put the, if you submerge the embryonic limbs completely in media, they die really fast. But if you culture them right at an air-water interface, so you fill the media up right to the surface of the foam, and seal the bioreactor so it stays, the humidity stays high, you can keep them alive. So Neve spent a lot of work figuring out optimizing all of the conditions to keep them alive. And we can keep them going for 5 to 6 days. After that, we start to worry about cell death. Yeah, great.
Very cool.
Okay, so it looks like, if I end the poll and share the results, it looks like blue wins it with 62% of the vote. Okay, so let's go to blue. All right, so here I want to tell you about this idea that we have that we call mechanotransductive learning. This is work that was done by a PhD student in my lab, Devin Mason, who's now a postdoc at the NIH. So Devin was focused on mechanobiology, and he came to my lab to ask this question, which is mechanotransduction a one-way street? So when I was brought up in mechanobiology, maybe this was my naivete, but I thought of mechanotransduction sort of as this monotonic progression from mechanical forces to cellular outcome, right? So mechanical cues activate the cytoskeleton, which causes information transfer into the nucleus and results in some change like differentiation. But what Devin was asking was, can we go both ways? And by that we mean, does this mechanical environment activate a feedback loop that can influence how the cell interprets these mechanical forces to begin with? Okay. So this brings me to back actually to when I was a kid, when I was reading the Choose Your Own Adventure books. Our next door neighbor had this car, a 1958 Chrysler Imperial. And Irma kept this car immaculate. White wall tires, always perfectly white. Everything was clean. And I loved staring at this car. But this car is actually really special because it, and I'll tell you in just a minute. I want to first introduce this guy. He's one of my favorite former graduates of the University of Pennsylvania. He got his mechanical engineering degree from Penn in 1912. And he was, at the age of 5, he was injured and went blind. But he went on to be a very famous inventor. In 1950, he got a patent for this thing that he called the speedostat. And some of you may be able to guess what this was. This, of course, was the cruise control. And the very first implementation of the cruise control in an automobile was this car, the 1958 Chrysler Imperial. Of course, Steeder's speedostat was an analog device, and now for cruise control we use a digital cruise control but it essentially functions in the same way. So let's say we're driving along at 50 miles an hour and we want to now go to 55 miles an hour. So we're going to increase the speed setting on the cruise control. And what you want is the smoothest and also fastest transition from 50 to 55. We could do an overdamped feedback system that leads smoothly to the new set point, but takes a long time. We could do one that's underdamped, where we get this overshoot that oscillates and also takes a long time to equilibrate. An optimal feedback system will overshoot a little bit, but then equilibrate. Okay, and so the question is, do cells in response to mechanical perturbations exhibit this kind of feedback behavior? So we sat down and we started to look at feedback loops that are induced by mechanical signals. And at short timescales, at timescales of minutes, there's this behavior called tensional homeostasis, where cells that get perturbed, like if you poke it with an AFM, the cytoskeleton will respond, but then it will equilibrate back to its original equilibrium within about 10 minutes. At long time scales, we have this behavior of mechanical memory, where if you culture cells on a soft or a stiff substrate for a long period of time, then you move them to a new environment, they will maintain an acquired memory of that mechanical history and behave differently depending on their mechanical past. But in between this time scale of days and this time scale of minutes, there's a time scale gap, there's a knowledge gap. And so here we propose this idea of what we call mechanotransductive learning, in which a cell has to respond to a set point change in mechanical environment. And the question is, do the cells exhibit this kind of overshoot and remodeling behavior? Now, we decided to study this in the context of cell migration, because there's lots of great pulling forces and pushing forces. And so the interaction between cells and the matrix is controlled at these integrins, right, and focal adhesions. And there's a feedback loop already that cytoskeletal tension can promote focal adhesion formation and maturation, which results in more cytoskeletal tension. But what we wanted to know was what puts on the brakes, and specifically, is there a feedback loop that can put the brakes on this? So the first experiment that Devin did was to just block transcription, block new gene expression altogether, or translation with this puromycin, and watch the cells as they migrate. So we use this wound migration assay as the cells migrate into a gap. And what we found is that control cells activate and then they reach an equilibrium speed. But if we treat the cells at time 0 with either a transcription or translation inhibitor, they will begin to migrate just fine, but then come to a rest. So here's a video of a cell treated with DMSO. And you can see that the cytoskeleton is highly dynamic and the cell is moving around. Now, here's a cell that we're going to treat at time 0 with actinomycin D that blocks transcription. And the cell is going to begin moving dynamically. But after we reach about 4 hours, you can see the clock at the top, the cell is going to begin to slow down, and the cytoskeleton is going to become much less dynamic. And this results in arrest of motility. So this suggests that somehow there's transcriptional events that get activated that feedback to modulate the cytoskeleton. So we propose that maybe YAP and TAZ can mediate this feedback. We find that they are activated in migrating cells. And if we deplete YAP and TAZ, we can abrogate the motility of these cells. So this suggests that there's a feedback loop. And just like we saw with the transcription inhibition, the depletion of YAP and TAZ caused over-maturation of the cytoskeleton and formation of focal adhesions. So you get tons and tons of focal adhesions, and the mechanical properties of the cell go up. So there's high mechanical stiffness in the cell because of this cytoskeletal tension, which suggests that somehow YAP and TAZ are regulating the transcription of genes that can negatively feedback to modulate the cytoskeleton. Now, at the time, there was some data suggesting that YAP and TAZ could regulate genes that control focal adhesion disassembly. And so that process happens by integrin internalization. So Devin came up with this experiment where he would hypothesize that YAP-TAZ depletion will decrease integrin internalization. And so what he did was he labeled live cells with a fluorophore to label the beta-1 integrin and let them begin to migrate. Then he washed the cells with an acid such that all of the labeled beta-1 on the surface would disappear, leaving only the internalized beta-1 integrin. And the hypothesis then is that there's going to be less labeled beta-1 in YAP-TAZ depleted cells. But what he found was that there was more endocytosed beta-1 integrin. And if we look at the presence of beta-1 integrin in Rab7 positive endosomes that are responsible for recycling integrin back to the membrane, there's no difference. So concomitant with an increase in the number of focal adhesions, we have an increase in the number of endocytosed beta-1 integrins. So it's not a failure of focal adhesion disassembly. Instead, we found that YAP and TAZ depletion caused an increase in myosin phosphorylation. And we could partially rescue the ability of these cells to migrate by inhibiting myosin. Now, in Devin's paper, he identified a couple different pathways where this can happen. I won't go through all of the details. You can find them in the paper. I want to come back to this idea of mechanotransductive learning. So the question is, do cells exhibit this overshoot and remodeling behavior on these sort of timescales of hours, right? So using live traction force microscopy with Amit Pathak at WashU, we found that cells, when you initiate the cytoskeleton, they peak, and then there's an overshoot and a remodeling to an equilibrium. But when we block this feedback loop by depleting YAP and TAZ, the cells will initially respond, but fail to have this remodeling. And this is what we'll call this feedback interrupted versus mechanotransductive learning. All right, not right now, bud. Okay, so if this is true, it should influence cell motility, right? So in control cells, we find that they start out very quickly, and they slow their motility as the cytoskeleton polymerizes. But as the feedback loop implements, they will accelerate back to equilibrium. In contrast, the transcription inhibited cells, or YAP-TAZ depleted cells, slow down and then arrest. So together, this suggests that these cells are behaving according to this feedback pattern that we're calling mechanotransductive learning. That is sort of similar to the cruise control in your car. Okay, so I want to finish by just giving an overview of the map that we've taken here. We started with the Fox brothers and their criminal advantage because of, at least until the fingerprint came out. And we talked about YAP and TAZ, which are these transcriptional regulators that respond to mechanical forces. And we found that if we delete them from skeletal lineage cells, that the mice die. So you all chose to look at the embryos, and we found that in these embryos, there's a defect in endochondral ossification. And if we take those embryonic limbs and we put them in a bioreactor, mechanical loading will promote this bone formation, but is abrogated when YAP and TAZ are depleted. And then finally, we showed that YAP and TAZ, when activated, modulate the cytoskeleton through a feedback loop that is consistent with this idea of mechanotransductive learning. So I want to thank everybody for your attention. There's other stories that we could have covered, but I look forward to conversations with all of you. Feel free to contact me if you have questions. I want to thank the folks in my lab who did all the work, our wonderful collaborators, and the funding that makes it possible. And now I'd love to be able to take questions.
All right, feel free to type your questions in the Q&A chat and I can read them off, or if you want to chat them directly to me, that would be fine too. I suppose while we're getting a few questions from our audience, I can start things off with just one. I guess it's more of an observation that I'm curious if you also noticed, and maybe you could tell me if it's meaningful at all.
Yeah.
When you were showing those images of embryos and the distribution of YAP and TAZ kind of around the ossification center, it looks like YAP and TAZ have different spatial distributions. I'm wondering if that's meaningful somehow.
Yeah, great question, Nick. Okay, so I think so far my answer is I'm not 100% sure. So if we look at that particular time point, we see these differences in distribution. If I look a couple days later, all of the cells everywhere are just cranking out YAP and TAZ. So is it meaningful? Well, it's possible. So Roland Baron had a study where they knocked out YAP out of osteocalcin expressing cells and found that they did it either YAP or TAZ, and they found that TAZ knockout affected the cortical bone and YAP knockout affected the trabecular bone more. We're still trying to figure out whether there's compartment specific differences here. It's really possible. So, you know, I think of YAP and TAZ as sort of this hourglass shaped signaling pathway where there's multiple things that affect upstream and multiple possible outcomes downstream, and it really does depend on context.
I guess if I can follow up on this question, it's an observation I also made. But have there been reports that either YAP or TAZ can act independently of each other in terms of transcription?
Yeah, great question. Okay, so the answer is yes, and it depends on context. So if you take a global knockout of YAP, embryo dies 8.5. Okay? If you globally knock out TAZ, they live to adulthood. They've got some liver problems. They've got some. They got a slight skeletal morphology. They have some other problems and whatnot, but they don't die as embryos, right? So clearly, in some contexts, one of them is really important and the other one is dispensable. And there's other contexts where it's opposite, where TAZ is really important and YAP is less important. Our finding is that YAP and TAZ act combinatorially in skeletal lineage cells. So you only see the perinatal lethality when you knock out both of them, and a single copy is capable of rescue. But there are distinctions. So we do see that TAZ knockouts are more, have a stronger phenotype than the YAP knockouts.
We also have a question from Chike Sao.
Yeah.
Would you be able to unmute her, Kristen?
Probably, let's see. So while you do that,
Chike, you should be able to go ahead and ask your question.
Okay, thank you so much. Can you hear me?
Yes.
Okay, so the YAP and TAZ, I wonder what kind of mechanical, how the mechanical or stimuli can activate the YAP and TAZ.
Yeah. Okay. So your question is, what's the mechanism by which mechanical forces activate YAP and TAZ?
Yes.
Okay. So I think, to be honest, the jury is a little bit still out on this question, though there's been a lot of activity and a lot of interesting findings. So there are observations that mechanical forces on the cytoskeleton, say actomyosin tension, cause deformation of the nucleus. And deformation of the nucleus opens up the nuclear pores, which allows YAP and TAZ to translocate in. Okay. And others, for example, Rob Mock from our group has found that if you interfere with the link complex and you prevent the cytoskeleton from connecting to the nucleoskeleton, you can stretch the cell all you want, but you don't get YAP activation. Right? So somehow the deformation of the nucleus is essential. And the first papers suggested that this mechanical activation was completely independent of hippo pathway kinase activation. But there's been other papers showing that there's an interaction, a signaling interaction between hippo and the mechanical activation. And so I think there's still something we don't know. And I would love to learn what it is. But
And
yeah.
another following question about it is that the nuclear localization of yapentase can be an indicator or biomarker about increased biomechanical loading or stimuli.
I really wish that was true. So we've done a bunch of experiments in vitro, and we see this beautiful interaction between and correlation between the mechanical stiffness of the matrix and the amount of nuclear localization, et cetera. But when you look in vivo, it's much more complicated, right? Because you got all these growth factors and it's in 3D, and you got the cytoskeletal organization of the matrix in reference to, of the cytoskeleton in reference to the matrix. We haven't been able to find a real clean connection between here's how much mechanical stress and here's how much nuclear yap. So I don't think we can use it as a readout or a biomarker, unfortunately.
Okay, thank you.
Yeah, great.
Got a few more questions, and I don't think we'll be able to get to them all, but we do.
Nick, if I may interrupt.
Yes, go ahead.
Maybe as people have to go to other commitments, if Joel doesn't mind, we've got about 4 questions that are typed up in the chat, and I see the people who asked them actually raise their hands. So I'm going to allow them to talk.
Great, that's great.
So let's go to Eric Small.
Okay, can you hear me now?
Yeah, go ahead, Eric.
Oh, awesome. So hippo signaling was sort of historically linked to regulation of organ size by contact inhibition and regulating proliferation, and mutations in yapentase break that link and cause sort of unregulated growth. What is the contribution, if any, of proliferation and contact inhibition to what you're seeing here?
Yeah, really good questions. So we're chasing this real hard right now. I really want to know, in, let's say, osteoprogenitor cells, how are they mobilized? Are they mobilized primarily by migration, or does proliferation play a role? Do yapentase regulate the motility and or proliferation of these cells? How is it influenced by the cellular context, like contact of one cell to another? So we're looking at this in the embryo. We're looking at this during fracture repair. We have a story that I didn't tell you where we found that yapentase regulate proliferation of periosteal progenitor cells after fracture. So we're real interested in these questions, but don't have complete answers yet. Yeah.
Thanks.
You can go to Antonio. Tony. Let's see. You should be able to speak, Tony, now.
Yeah, there you
Oh, perfect. Can you hear me?
Yeah.
Great. Hi, Dr. Boerckel. Amazing presentation. So my question is, were the osteoprogenitor cells used in the in vitro experiments where you did silence the yapentase from the paper Mason et al. 2019?
Yeah, great question. Okay, so all of those data that I showed you were in endothelial cells. We have some data in the paper from the knockout mice where we show that they also have, if you knock out yapentase, they have this elevated cytoskeletal tension. But we've not yet teased out all of the little pieces, right? Does the feedback loop work the same way? Are the dynamics the same, et cetera, et cetera? So we're working on this right now. But the cell type is really important. So there are some cell types where yapentase act as co-activators, and some cell types where they act as co-repressors for the same genes. And this is a question that the field has not yet answered. And I honestly have not seen anybody working on this. Why do yapentase sometimes act as activators and sometimes act as repressors? I would love to try to figure out the answer to this question, but it's a really hard one.
Yeah, definitely.
So we're trying to think through how we could answer it.
Thanks. And more theoretical. So do you think if these osteoprogenitor cells were used for such a study, would it matter at what kind of embryonic stage those cells were isolated and utilized? Do you think there will be like different results and outcomes by silencing this yapentase at different stages?
Yeah. So we're also working on this a little bit. So in a slightly different way, but maybe we'll be able to address it. So we think it's going to be important to study the appropriate cells when we go chase after the cell biology. So for our fracture healing studies, we're isolating these mesenchymal progenitors from the periosteum after their amplification shortly post-fracture. And for the developmental biology studies, we're taking cells from the embryonic limbs. And there are similarities, but also differences between these cell types. So it is possible, because of the transcription factor milieu in one cell type versus another. If they have different transcription factors, then YAP and TAZ could have very different functions in one cell type versus another.
Perfect. Thank you very much. It was great.
We will go to Qialong.
Hey, Joe, can you hear me?
Oh, yeah.
Thank you so much for the nice talk. Very, very inspiring every time when I hear your talk. I guess I have two questions. So the first one is for the embryonic stage. I think you show 14.5 or 15.5, right? So in terms of YAP and TAZ expression, do you see even early on, do you even in mesenchymal condensation at that stage, do you see that upregulation of those two genes?
Yeah, good question. We haven't gone that early yet. So, recently we've started looking at expression levels by in situ hybridization approaches. And what's interesting is if we look at the protein level, we see lots of YAP-TAZ protein in these cells during development, but not a ton of RNA. Compared to, if you look for collagen 1 or collagen 2 in the right cells, they're just cranking out those genes. But YAP-TAZ expression at the mRNA level is pretty low. So we're trying to figure out, what's the best way to get these dynamics starting from the very beginning of the limb bud to later on.
Interesting. So my another question is kind of end of the spectrum. So like aging mice, like if you have an inducible knockout, either YAP or TAZ, what is your speculation like? If you knock out the later stage of the mice, do they get easier to get OA or not?
Yeah, we haven't done any aging studies. We have done an inducible knockout in fracture healing, where we let them grow to skeletal maturity and then fracture, and then compare what happens to fracture healing if you have a constitutive knockout where you knocked them out from the beginning of development, or the inducible knockout where you knock it out right before the fracture. And there are differences, right? So YAP and TAZ play a role in the development of the periosteum, which means the fracture healing process is affected. But we haven't taken them into aging.
Very interesting. Thank you very much for your talk.
Thanks.
We have a couple of questions in the chat. One from Sarah Van Houten. And she was asking about if in the bioreactor, if you thought about incorporating different stimuli experienced during development, including electrical stimulation, which plays a critical role in muscle development, possibly study the synergistic effects of those stimuli on development.
Yeah, that's a really interesting question. We haven't done anything with electrical stimuli, though. I don't know if you're familiar with Daniel Cohen's work from Princeton. He's been studying electrical stimulation and control of cell migration, and his videos are mind-blowing. And it's sort of I had sort of dismissed electrical stimuli as a really important signal for a while, but after seeing his stuff lately, it's really remarkable what he's doing. So I think it's really possible that there could be interactions between those electrotaxis and durataxis, et cetera.
And then one last question from Wasil Lee. She was asking you about your perspective on the differential role of YAP-TAZ morphogenesis and actin filaments in osteo versus chondral, the differentiation and the differentiation.
Yeah. So this is one that we're really interested in figuring out in as sort of the next big questions we want to figure out. Because we see different responses of chondrocytes versus osteoblasts to mechanical stimuli. And in fact, you know, in all of our experiments so far, when we mechanically load chondrocytes, we see a downregulation of YAPTAZ activity. And in osteoblasts, the opposite goes up. So maybe this is a function of the cytoskeletal architecture, right? The three-dimensional spherical organization of the chondrocytes versus the surface-bound, you know, polarity of the osteoblasts. I don't yet know. It's also true that they have different transcription factors, right? There's totally different transcription factor milieu in an osteoblast versus a chondrocyte. So there's lots of really interesting questions here. And depending on your perspective on this idea of hypertrophic chondrocyte transdifferentiation, there may also be interesting questions about how do you go from one to the other, right? Thanks so much.
I don't think we have any additional questions. Do you see
Right.
anybody else, Nick?
Nope, I don't see any other questions.
Right. Well,
I just
thanks,
want to say,
everyone.
yeah, thanks for organizing this, Kristen and Hanny. And thank you to our speakers so much. Excellent talk, really interesting stuff, and great discussion, everyone.
Thanks everybody for all the questions.
Excellent talk. Really enjoyed it. And thank you, Nick.